back to what we were talking about prior to that. We had talked about active transport and how active transport was basically moving things against the concentration gradient, and it always required energy. The example that I gave you that every cell has, that's probably the most important example, textbook example, was the sodium-potassium pump. The sodium-potassium pump would pump 3 sodium ions out, bring 2 potassium ions in, and it needed an ATP to do that. The ATP was what sort of caused the protein to change its shape to move everything. So one of the things, and this is sort of related, one of the things that happens when you have a pump like the sodium-potassium pump is that it can generate something called membrane potential. Membrane potential is a situation where you actually have an uneven charge on 2 sides of a membrane. So it's sort of like if you think about a battery and how a battery has a positive and a negative terminal. There's voltage in there, and it's sort of stored potential energy. When that battery is hooked up to something, then the energy is released. This can actually be something your cell will do as well. Your cell can have a situation where it creates a charge, a voltage difference on one side of the membrane versus the other. And so that's called membrane potential, and we'll talk in a second about what it's used for. Secondly, very similar definition. A lot of people mix these two definitions up, but there's a different word for when you have a difference in the amount of ions, in the amount of chemicals, and the amount of charge. It's called an electrochemical gradient. So electro, for having an electrical difference, meaning the charge is different on 2 sides of the membrane, and then chemical because you also have a different amount of solute on the 2 sides of the membrane. So there's 2 forces that cause this, like I said, the chemical force as well as the electrical force. But again, a lot of people mix up these 2 definitions. So if they say something just specifically about there being a difference in charge, the name of that specific difference is called membrane potential. But the whole entire setup where you have a difference in charge as well as different numbers of solutes is called an electrochemical gradient. The sodium-potassium pump actually causes this situation because if you remember, it is pumping 3 sodium ions out and at the same time bringing only 2 potassium ions in. That means that outside of the cell, so this is our little cell membrane here, that would mean that outside of the cell is going to end up, assuming there's other ions around as well, but overall, you end up with an overall more positive outside than inside. Inside the cell ends up being more negative than outside because of the fact that you're pumping out more positive charges than you're bringing in. So this creates what's called an electrochemical gradient. This electrochemical gradient is the basis for a lot of things, but one of the main things that it's important for is nerve impulses. Your nerve cells normally are negative inside compared to outside. About negative 70 millivolts more negative inside than outside. When a nerve gets stimulated, what will happen is gated channels open, not the sodium-potassium pump, but other channels that are normally closed. They're called gated channels. So they have like a little gate like that that's closed. Normally, sodium ions are sort of stuck out here, so they're getting pumped out. When the gated channels open, like you touch a hot stove and the nerve gets stimulated, this opens up, and because there's a difference on the 2 sides, a bunch of sodiums will flow in. Because positives attracted to negative, and because you have more outside than inside. So sodium flows in, and that's the basis for how nerve impulses travel. It causes a whole chain reaction that when this gated channel opens, it causes the one next to it open, which causes the one next to it to open, and you end up with a impulse that will then travel to your spinal cord, which will then send another impulse back to tell you to move your hand away from the stove. And this happens so quickly that you don't have to think about it. If you really think about it, you probably have never touched a hot stove and left your hand there long enough to think, oh, this really burns, let me move my hand. It happens before your brain even processes what's happening. So these impulses travel very quickly. And a membrane potential? Yeah. So the difference between the two is if the definition just talks about the charge only, they wouldn't mention it's caused by ions, but they wouldn't mention ions. So if it just says there's a difference in charge on two sides of the membrane, that's called membrane potential. If they say a difference in ions and charge, if they mention the ions, then it's an electrochemical gradient. Yeah. So like I said, one word's just a little more, and I do find that people mix them up because they are very similar, really. This next slide. So any pump that causes membrane potential is called an electrogenic pump. So that would mean the sodium-potassium pump is an example of an electrogenic pump because it sets up that situation where it creates the voltage. And again, the voltage created by the sodium-potassium pump is more positive outside and more negative inside because it's moving out 3 sodiums and only bringing 2 potassiums in. Another electrogenic pump that's important is what's called a proton pump. Now, proton pump kind of confuses people because if you were going to see it in a drawing, you would not see the word proton. And I'll show you a picture of this on the next page. But what they would actually show you, and these require energy also, so you would have ATP involved. That's sort of the way they represent energy. But if this was a cell membrane, here's our phospholipid bilayer here and here. What an electrogenic pump would actually, what it would show, a proton pump, is it would be pumping hydrogen ions. Why? Because, if you remember, hydrogen's number 1 on the periodic table. It has 1 proton and 1 electron, which means that hydrogen ions, meaning you took away the electron, are basically nothing but a proton. So they call it a proton pump, but what it really is pumping is hydrogen ions, just to be clear, which in essence are a proton, but a lot of people don't make the connection. They think proton pump, they're going to see some picture that has the word proton or drawings of protons. It's not. It's going to have a drawing of hydrogen ions. That's what it's technically pumping, because a hydrogen ion is a proton. Again, by creating this gradient, it actually creates a setup. It's sort of a potential energy setup that then when gated channels open, it can cause these chain reactions that I mentioned before. So again, 3 vocabulary words there: electrochemical gradient, membrane potential, and then electrogenic pumps. Okay, this is a picture of a proton pump. So this is what you would actually see if they were showing a proton pump. It would be hydrogen ions that would be pumped. Again, you see this in bacteria, but also in mitochondria, which makes sense because remember endosymbiotic theory, that mitochondria were once bacteria. So it makes sense that the kind of pump they would use would not be the sodium-potassium pump, but a proton pump like what bacteria use. All right, this next slide, I'm going to send out an announcement to remind people of this, but this is not on your test. So the whole section of notes about nerve impulses, I was just going to give you a little introduction to it, and you can write down the information if you would like. But there will be nothing on your test about this slide, there will be nothing about this picture, and there will be nothing about this slide either. And I know that if you're using the outline, it includes this picture. So this will not be on your test. I'm just going to briefly go over it. I mentioned this a moment ago, yes.
Is there anything on these slides that can help us in any way about the other information?
No, it's just like nerve impulses are a specific example of where the sodium-potassium pump sort of sets up a membrane potential, and then an impulse travels by gated channels opening. But honestly, there's nothing here that will help you further understand the other stuff. Yeah, and we're going to come back to this when we do a different chapter and talk about it again. So for right now, again, you can fill it in, but there won't be anything tested on it. So what I mentioned to you that in a nerve cell, which is usually long and skinny, it has a part at the end here, it's more positive outside than inside, and that's because of the sodium-potassium pump. So we think of our outside as positive, our inside as negative. When it gets stimulated, it causes sodium channels that have gates on them. They're so normally closed. They open. Sodium basically comes pouring in and makes the inside of the neuron now positive. that triggers the channels next to it to open, which triggers the channels next to that to open, and the whole thing basically travels down the neuron to the end, where neurotransmitters will take the message to either another neuron, to the spinal cord, to the brain, wherever that neuron might travel. But the bottom line is, the channels, these gated channels open, that makes the inside positive, and then the channels close. Sort of like if I open this door to my classroom, it doesn't stay open. It sort of slowly slides closed. The same thing happens here. So these channels open and sodium comes flowing in when the nerve gets stimulated. Like I said, you touched a hot stove. But then it closes. And once it closes, two things happen. Potassium channels open that let potassium go back in, and the sodium-potassium pump starts to restore things back to normal. So eventually what will happen is this will go back to normal again, and the nerve can be stimulated a second time. But I would comment, this would explain, because this does take some time to reset. So you stimulate the nerve, like if I smack a table really hard, you know, if you smack it, you know that you feel that pain in your hand stings. If you touch your fingers, they may actually feel numb for a minute or two. And you can't force them to feel anything. Because the nerve got stimulated, the pain receptors, it sends a message, and this whole time while this is going back to normal, the nerve can't be stimulated a second time. So it's almost like those nerves are sleeping temporarily. And so you wouldn't feel anything until everything resets. Then if you smack the table again, you would feel it. So that's in essence what's happening in a nerve impulse. Again, nothing on the test, and that's why I'm going through it very quickly. I'll explain it better and in a little more detail when we come back to it another time. This is what a neuron would look like, though. This area on the left side, this is where the dendrites are, where you would actually touch the hot stove. This would be the part that would be in your fingertip. And then it would carry this message, like I said, with the gated channels opening, and sodium flows in, which causes the next section to open, which causes the next section to open, and the whole thing just goes like a chain reaction. And then over here would be your spinal cord, where the message would then get translated to move your hand away. It would send a new message back to the muscle to pull your hand away. And you wouldn't have to think about it. You might not even realize what was happening until afterwards, and then you'd realize, wow, my hand really burns. But that would be after. And this diagram, again, is sort of a walkthrough of all the channels and everything, but again, you're not responsible for it on the test. But I like this part here because it kind of shows you what's happening. So normally the membrane is mostly negative inside. When those channels open, like I said, you touch the hot stove and all these sodium gates open, all the sodium ions flood in, and it makes the inside positive. That triggers the chain reaction to open the ones next to it, etc. And then as those doors close back, the gated channels close, it sort of goes back to normal, and now it's back, it's what's called resting potential, the resting state of the neuron. So, again, we'll talk about it more another time, but that's just sort of a very quick overview. This is actually the very last slide. Co-transport. So remember proton pumps I mentioned a moment ago that they pump out hydrogens, and remember how the sodium-potassium pump pumps out sodiums. Sometimes one of the reasons why these pumps are really important is because they are linked to other proteins. So if you look at the picture, this down here, this is a transport protein that brings sucrose into the cell. Now sucrose is sugar, so that's something the cell probably needs in order to survive. But the problem is that sucrose can only be transported across if hydrogen also attaches to it. It's sort of like if you've ever been to a carnival and you wanted to ride something, but that ride doesn't take single riders. So you had to wait until you found somebody that could partner with you. That's what's happening here. So sometimes what will happen is, in your cells, you have proteins like this sucrose co-transporter, where and this is called co-transport, where that protein brings in something that the cell needs, but it'll only transport it if some second molecule also binds. So if you were asked, does this molecule here require energy? It does not. There's no ATP here. Sucrose naturally flows in this direction. The problem is, since it requires hydrogen, and the only way the hydrogen ions get out is through this pump, it actually does indirectly require energy. Because if this proton pump shuts down, there won't be any hydrogen out here, and that means this sucrose transporter can't bring sugar in, so the cell would die. So co-transport, that's, like I said, is where you have a protein in the cell membrane, and we have some of these in our cell membranes as well.